Thank you for standing by and welcome to the Arovella Therapeutics Investor Webinar and Q&A today, discussing the company's announcement this morning that it has licensed the rights to a novel monoclonal antibody /CAR to treat cancers. All participants are in a listen-only mode. There will be an initial slide presentation lasting for approximately 20 minutes, followed by Q&A based on questions received via email and those that have come through live. If you have a question you'd like to submit, please feel free to type it in using the Q&A function within Zoom. Appearing on behalf of Arovella Therapeutics today, we have the CEO and Managing Director, Dr. Michael Baker. Michael, I'll hand it over to you.
Wonderful. Thanks, Matt. I've got a little bit of feedback. Can you hear that? That's better. And thanks very much for putting on the webinar to the team at NWR, and of course, a big thank you to everybody that's taken the time to listen in today. And we are particularly pleased with what we've announced this morning. I'm certainly looking forward to taking you through the story. And what it is, it's an exclusive global license for a unique monoclonal antibody target. It's a DKK1 peptide, which I'll certainly talk more about in a moment, but it comes out of the University of Texas MD Anderson Cancer Center, which is well regarded as one of the best cancer centers in the US and if not globally. So we're particularly excited by the therapy, how it fits together with our existing platform. And again, thank you very much for dialing in. There we go. And there's the usual disclaimer. And just to jump straight into the license itself and the technology. So as I mentioned, it is an exclusive global license, which is wonderful for Arovella. We don't have any limitations on the disease target either. So it is across at this stage, we're just looking at cancers, but we're not limited to any particular type of cancer, which is great. And as I said, it is a license to a novel DKK1 monoclonal antibody that targets a peptide of that protein that's found on many different cancer types, including blood cancers and also a range of solid tumors. And what we've seen in the literature that's been published is that it's described as something that's really overproduced in many different cancers, which is what sets it up to be useful for those particular cancer types. And that we think really validates the use of this technology as a cancer target, particularly for what we're doing with our platform. And in terms of the development, it's been under development for quite some time. Firstly, initially developed at the University of Texas MD Anderson Cancer Center, and then more recently, the inventor was recruited to the Houston Methodist Hospital, where he's continued to research and really apply the technology into the chimeric antigen and cell therapy space. And so, as I said, it's a monoclonal antibody with chimeric antigen receptor potential, and there's good data to suggest that it could work alone as a monoclonal antibody. But again, the more recent data that we've got from the Inventor about using this as a chimeric antigen receptor for cell therapy is very compelling, and is certainly what has driven us to get involved with the technology. But also, it's exciting because we do see that there's good overlap and synergies with our existing platform. And in terms of the deal architecture itself, so we can well funded to cover the upfront license fees, which is great. And of course, like most biotech deals, it's got standard milestone payments and royalties. So as we succeed, we share the success with MD Anderson. And in terms of intellectual property, which is a key parameter for technologies in this space, because it essentially becomes the currency, there's very clear patent protection with both composition of matter and method of use claims. It's got long patent life out until 2039. It's been filed in major territories, and we've already received a favourable search report and don't expect any issues with freedom to operate, which is very pleasing. In terms of Arovella, so as of pre-open this morning, we're valued about just under 17 million. We don't think that reflects the value of, firstly, the iNKT cell therapy platform, or certainly the technology that we're discussing today. But we do have cash in the bank of 5.1 million as of end of September this year. So we're not actively looking to raise money. In terms of our ownership, we do have a number of good small institutional and high net worth individuals at the top end that are quite supportive and understand the investment horizon, but also the returns that can be achieved in this space. Certainly, we would like to have a little bit more concentration at the top end, but again, we're very keen and looking to welcome investors that are very familiar with an investment in the biotechnology space, the investment horizon, the risks, but also the returns that can be achieved in this space. And just jumping into the technology and why we think it is unique. So as I said, the DKK1 monoclonal antibody was developed at MD Anderson, and the inventor's nicely shown that it can be incorporated into a chimeric antigen receptor. And what's very clear in the scientific literature is that DKK1 is increased in many different blood cancer types. Multiple myeloma is one, which is a form of blood cancer, a pancreatic cancer, which is a very nasty, difficult to treat cancer, breast cancer. He's shown data with triple negative breast cancer, which is quite exciting, and also lung cancer. And what's also quite clear is that, yes, it's overproduced in cancer, but also what has been seen is that it leads to poor overall survival and also shorter disease-free survival, again, which is why we think this is an important target to tackle in particular cancers. And it really extends the limited set of targets that we have, both for blood cancers and solid tumors, which is quite nice. And we believe it is differentiated from others by the fact that it does span so many different cancer types in its presence and overproduction. And what the inventor's been able to show really nicely is that when he puts it in T cells, and I'll talk more about that here. So T cells being the conventional cell therapy that now have 5 approved. And what I've got sticking up on the right here, this would be the chimeric antigen receptor for the DKK1 technology. He has shown in animal models for multiple myeloma, pancreatic cancer, lung cancer, and breast cancer that it does extend lifespan, and it is looking quite compelling. And so really what Arovella intends to complete is to take the DKK1 CAR and put that into our iNKT cell therapy platform and use that to tackle particular diseases. And first off the rank, we're likely to tackle multiple myeloma, but of course there is potential to go after some of these solid tumors as well. So just to give you a bit of an overview on the different cancer types for which this has shown activity. So multiple myeloma, as I said, it's a form of blood cancer for which there's still no cure per se. It affects just under 35,000 patients per year, and still a pretty poor survival rate at 1 in 2 after 5 years. Lung cancer, where it's a very prevalent condition at just over 235,000 cases per year in the US, and still a pretty poor 5-year survival rate. Triple negative breast cancer, which is a form of breast cancer that's particularly aggressive and prone to spreading and also to recurring. Again, not an insignificant market size at just under 30,000 cases per year. The survival rate looks high here, but this is a little bit unclear, because the moment the cancer does spread throughout the body, the 5-year survival rate does drop markedly. So again, a very important disease to tackle. And pancreatic cancer, as I mentioned, it's the 4th leading cancer killer, and it's around 60,000 cases per year in the US alone, and the 5-year survival rates is dismal. And so really that's a cancer type that it's dire that we get more therapies to treat that particular form of cancer. And just to talk a little bit more about the data, because I think that's ultimately what's really important about these types of technologies. This is looking at using the DKK1 CAR-T cells and looking at a cancer called multiple myeloma, which is the form of blood cancer that I referenced a moment ago. And what the inventor's shown here is it's using the technology in 3 different types of multiple myeloma models. So the 1st, 2nd, and 3rd represent the different model types. And this has been repeated a number of times for each. And what we're looking at here is on the vertical axis, how long the animals, or how many animals are actually surviving. So once that gets down to 0, that means all the animals have succumbed to the disease. And on the horizontal axis, we're looking at how long they've survived for. And remarkably, in all 3 of these models, we see that between 30 and 40 days, if the animals aren't treated, which is the black line for each of the models, all the animals die. Whereas if we treat them with the DKK1 CAR-T cells, we see that they have at least out to 50 to 60 days for the length of these experiments. The mice are all still alive. And again, why we're excited about this specifically because of the iNKT cell therapy platform that we developed. Multiple myeloma also produces something on its surface called CD1D, and our iNKT platform will already recognize those particular cancers that have that on their surface. And so when we combine the DKK1 CAR with our iNKT cell therapy platform, will have two ways to attach to and kill the cancer cells. So we think it will be more effective, but all that while being a product that we're developing to be used off the shelf so we can start from a healthy donor. So hopefully we've got a more effective product that can be used more broadly and more cost effectively. And to talk more about the iNKT cells, it is important. So what we're talking about in terms of this type of therapy, it is a cell therapy. And what we might liken that to is a 21st century blood transfusion, where in our case, we'll be taking blood from a healthy donor. Now, within that blood sample, we've got many different types of cells, but for our purposes, we're interested in the immune cells. And there are many different types of immune cells. So I've listed a few here: macrophages, natural killer cells, and T cells. As I said, our platform of interest is the iNKT cells, but we might consider all these immune cells as the soldiers of our immune system. And so they go around looking for things that don't belong, and their job is to trigger the removal of those components. And so why we're particularly interested in our iNKT cells is because they are very potent. And that's why we've actually depicted this one here as an armed soldier, because it can already find, attach to, and kill cancer cells. So as I said, iNKT cells are one of the most potent naturally occurring immune cells. They already naturally target and kill cancer. And what they do when they're activated is they actually bring in the other immune cells to assist in removing what doesn't belong, in our case, cancer. And nicely for us, as well as I mentioned, because this, what's called an invariant T cell receptor on the surface, doesn't change between these cells, between people, they can actually be used from one person to the next. And so that's what we call off the shelf. And that is important because these cells don't cause graft versus host disease, which is a potentially fatal outcome for other types of cell therapies like T cells. And more importantly, when we combine iNKT cells with CARs, for those cancers that produce the protein CD1D, we actually see that they're more effective over conventional cell therapies. And so just to give you an overview of how it actually works in a cartoon picture, we've got our iNKT cell. It has its invariant T cell receptor that can already find and kill cancer. We engineer the CAR-INKT cells to express or produce the CAR. So in this case, it would be the DKK1 CAR. And then when we give those cells back to a patient, what actually happens is they'll find the cancer cell, the ITCR will find what's called CD1D on the surface. The CAR finds it's called an antigen. And in the moment that happens, the iNKT cell releases all these components that essentially cause the cancer cell to die, all the while recruiting other components to assist in the fight. And so that's what we've seen already for our lead product, ELA-101. And I won't talk about this in great detail, and there's a lot of data here, but it's just to highlight the key features of the platform. We are developing ELA-101 as an off-the-shelf therapy for cancers that produce CD19. And we've already seen that the data for this therapy is very compelling for cancers that produce CD19. And I won't go into too much detail, but what we see here, this is after 3 days of treatment with our mouse. Anywhere there's color indicates cancer. And in our technology, you see that there's no cancer cells present. So we know that our therapies are more effective at removing cancers that have CD1D than conventional cell therapies. And it also, when we see it long term, it actually then translates into better survival of the animals as well. So this original data was from Imperial College, but it certainly validated the use of the platform. And that's what we're hoping to see with this next CAR as well. And for that particular program, we're getting close to selecting our manufacturer for the product, which we'd be using to develop into clinical trials. Now, in terms of the data, I think for the activity is incredibly strong. And so it's important that we also are comfortable with the safety of this program. And the inventors already demonstrated that for the DKK1 CAR-T cells, they only kill the cells that have the target on their surface. He's put them with healthy blood cells and doesn't see any impact on those at all. But when administered to mice, there's no weight loss or anything that suggests that the cells themselves are causing any issues. And in terms of looking for off-target effects on different tissues, he screened 35 different tissues and found that only one, which is the tonsil, was non-specifically targeted by the monoclonal antibody. But nevertheless, now that we've licensed the technology, what we'll be confirming is that it doesn't target or attack any healthy cells and only goes after the cancer cells, but also will combine the DKK1 CAR with our iNKT cell therapy platform to see if we get that that increase in activity against the particular cancers of interest. So as I said, the inventor himself, Professor Qing, he's an incredibly well-credentialed individual. He's medical immunologist, so he's an MD by background. He's got more than 25 years of experience working with immune cells and different cancers, but certainly considered one of the leading investigators in the field of tumor immunology, particularly looking at multiple myeloma, but also other cancers. So very, very strong pedigree, originally training at the Karolinska Institute in Sweden, before MD Anderson moving into the Cleveland Clinic, and now he's at Houston Methodist. He's also takes on two pretty high profile positions as director for the Center of Translational Research for Hematological Malignancies, and he's also the associate director for the Center of Basic Research Programs at Houston Methodist. Now, I won't go through the very long list of grants that he has been awarded. It's too many to name, but where he is now, Houston Methodist, he was recruited to that institute on what's called a CPRIT grant for a total of $6 million to fund his ongoing research, which has been used to fund some of the research for this therapy. And he's published more than 160 articles with his colleagues. So again, just a very well-credentialed individual, and it's a pleasure to be discussing the technology with him as we move forward. And as I mentioned, patent protection is critical for these types of therapies. And as I mentioned earlier, there's patent life out until 2039. This is the first DKK1 CAR product, so it has robust patent protection. And again, it's been filed in key territories: US, Europe, Canada, China, and Australia. But already we've received a favourable search report, and preliminary FTO analyses don't find anything that will get in the way of this getting granted in those major territories. And in terms of the commercial landscape for iNKT cell therapies, it is certainly worth looking at, because in the cell therapy space, there are many companies in the T cell and natural killer cell arena, and that's dozens, if not hundreds, of companies. But for the iNKT cell therapy space specifically, we only see that there's 4 companies today. So it's a much smaller, more niche area, it's wonderful that we find ourselves in such a unique spot. But quite interestingly, over the last 6 months, we've seen a fair bit of commercial activity for each of these players. APPIA Bio came out of stealth essentially in May, raising 52 million in their Series A. And just in August this year, they entered into a strategic partnership with arguably the world's best cell therapy company, Kite Pharma, for a total deal value of 875 million. And MINK Therapeutics, I should just point out actually APPIA Bio, so they are preclinical, not looking to get into the clinic until late 2023. MINK, they listed last October now. They're phase 1, and they raised 40 million and currently valued at about just over 200 million. And Qing Therapeutics also had a commercial outcome in May 2021. They were bought out by about a $500 million company at the time, Athenex, for about 185 million in total deal value with cash equity and milestone payments. And of course, Arovella, I mean, it's not hard to see we're valued considerably less than the others, but over the course of this year, we've acquired what we think is a really exciting platform. And of course, now we've just added to that platform through the acquisition of the license to the DKK1 technology from MD Anderson. And so moving forward, we've certainly got a lot of work ahead of us, which is we're certainly looking forward to getting underway and working with both Professor Qing and Professor Tassos Karademetri from Imperial to develop this into clinical trials. And in terms of the next steps, as I mentioned, the first things to do for Arovella's end is to confirm the DK1 technology specificity in terms of targeting cancerous tissues, but also to combine it with our iNKT cell therapy platform and to initiate the preclinical studies with the iNKT cells for various tumor types, initially with blood tumors, but blood cancers, but looking to potentially move into solid tumors. And as we progress that work, we'll enter into a sponsored research agreement with Professor Qing to get him and his lab to develop or to broaden the IP as well, which is important. And hopefully before the end of next year, we're completing the preclinical studies for the relevant tumor types, looking to initiate process development for manufacturing, and really developing and refining the drug development, the drug product manufacturing strategy. And in terms of our existing iNKT cell therapy platform, so a really key focus is to continue to analyze the landscape to find new CARs. So we've been looking probably for the last 18 months, and there are quite a number of interesting things that we may consider moving forward with. We'll also be continuing to prosecute the patents for the platform. We're very close to selecting our manufacturer for the iNKT cell therapy, which we expect before the end of the year. And that will trigger in the first half of next year the production of the plasmids that go into the cell therapy product. And of course, we'll be looking to recruit additional cell therapy experts as well. And so for the second half of the year, we'll be aiming to complete the manufacturing of the plasmids, which is one of the components that goes into the therapy, before initiating the manufacture of two more components, the lentivirus and iNKT cells for clinical trials. And at that point, we'll be aiming to finalize the clinical trial plan for the therapy to take into phase 1. And so that's our cell therapy pipeline to date. We have 2 major products. Firstly, the ALLO-101, which we're using to target CD19 expressing lymphomas. As I said, the key next steps for that are the manufacturing to take into phase 1. And ALLO-104, which is the CAR now that's going to target the DKK1 peptide, which we're developing for multiple myeloma and potentially solid tumors. And we do have data for earlier stage CARs for cancers and different disease states as well, but we're leaving those as undisclosed as they're quite early stage at this point. And in terms of our leadership team, I think this is something really important. Over the last 4 or 5 months, we've really built out some key roles, I think, in terms of our board, so our leadership, but also our management group and our scientific advisory board. So I don't think we need to introduce Paul in too much detail. He's got a very strong track record in the biotech space. For myself, for those of you that don't know me, I'm a former researcher, so I have a PhD in biochemistry before moving into drug development and then venture investing before joining Arovella in January last year. But I want to highlight 2 individuals, Dr. Deborah Barton and Dr. Liz Stoner. So first, Dr. Barton, she's a phenomenal individual, medical doctor in oncology by background, but very quickly moved into large pharma at Novartis and Celgene. And she spent the more recent parts of her career at well-known cell therapy companies like Iovance. And now she's the Chief Medical Officer of Charisma Therapeutics, an absolute superstar individual, and very happy that she's on our board. And same for Dr. Liz Stoner. Again, just a very broad background. So formerly an assistant professor at one of the US's best universities, Cornell, before moving into large pharma and in senior clinical development roles. And more recently, she's been at, and she still is, an executive partner at MPM Capital. So it's a well-known healthcare specific venture capital fund in the US. But really importantly, Liz was also the interim CEO of a company called Arovella Therapeutics, which went on to be acquired by another company, Vertex, for $950 million US in cash. So again, wonderful expertise, and it's an absolute pleasure to have these 2 individuals on our board. And of course, we're pleased that we've got obviously David Simmons, who brings a very important angle of corporate governance and compliance, but also David Phillips, who again, a really strong track record across the life science industry and all biotech, big pharma, but also venture investing at his time at SR1. And so I'd just like to stop there and just summarize, I think, the therapy, but also how things are moving forward for Arovella. So we're very pleased with the exclusive global license for the technology. It is unique. We see that there's a lot of potential to target blood cancers and solid tumors. So we're quite excited by the license and the technology. And again, all that comes from scientific literature and support and data that suggests this is a sensible target to treat cancer. We're happy with the deal terms of the license. Again, it's nice that we can fund the upfronts through our cash reserves. And again, happy that we can share the success with MD Anderson as we progress through to key milestones for the development of the technology. And I think importantly, as I've mentioned, the IP here is so clear, which is great. And that stands because of the uniqueness of the technology and the fact that nobody else in the world is working with a DKK1 CAR. And just onto the company. So I think we have an excellent leadership group, which is expanded rapidly over the last 6 months, as I said, to include also senior management people and a scientific advisory board. So we really think we're positioning the company and building excellent foundations for the company to move forward. And with that, we do believe we're on a solid growth trajectory, with now Arovella being the only ASX listed company that's developing an iNKT cell therapy for cancer treatment. But importantly, we're also the only company worldwide that has a DKK1 CAR. So as I said earlier, we're going to continue to look for unique assets that we think fit with our iNKT cell therapy platform. And we're certainly looking forward to progressing with some of those as we move forward. And just with that, I'd just like to say again, thank you everybody for taking the time. It's wonderful. I know everybody's got a busy schedule leading up to the end of the year. It's great to have you on the call, and I'd be very happy to take any questions.
Thanks very much for that, Michael. As you mentioned, we'll move on to the Q&A session. Again, as a reminder to the attendees, if you have a question, please feel free to enter it using the Q&A function within Zoom. Michael, the first question's I guess a broad but simple one. Why does today's announcement fit with Aravella's strategy overall?
Yeah, it's a great question. I think for us, it was the strategy really stemmed out of the technologies we're looking to acquire, which was in the immuno-oncology space. And since acquiring the iNKT cell therapy platform from Imperial College London, we have the platform, because that's the cell therapy, that's the cell type, which is the cell therapy platform. And now what we're looking to do is build, I guess, an IP or ring fence our platform by having more IP with different technologies. And that's what we see here today, that the DKK1, we think it's a very exciting target to treat cancer. Nobody else in the world has it. We have the IP for that. And as I said, as we continue, the idea is to continue to bolt on additional technologies that we can use to develop to take forward.
Thanks, Michael. The next question is, will you be looking to license any further assets?
Yeah, I think just to that point exactly. So over the last 18 months, looking for new technologies, we've found that there's a whole host of things that are of interest. And I think now it's about managing appropriately what we think fits best, where we see there'll be synergies with our platforms. But of course, if there are things that sit outside of our platform, we can also entertain that and have a dual or even triple platform approach in that respect as well. But that certainly will be a key feature. And again, looking at the team, Liz, myself, David, even Paul, all with great expertise in sourcing, evaluating, and acquiring new technologies. It's important that we're putting that key skill that we have, I think, to use.
Thanks, Michael. The next question is, how advanced does the portfolio need to be to attract industry funding as per one of the slides you presented? And is this something that you would plan to target?
No, it's a great question. And I think APPIA Bio probably, actually there's a few now. APPIA Bio entered into a strategic partnership with Kite in, I think, August this year for 875 million total deal value. So that's going to be upfronts and milestones. They're probably, we don't know exactly, but just under 2 years from reaching the clinic. So they're at a similar stage to us in terms of development timelines, or probably even a bit earlier. And there was another company, Shoreline Biosciences, I believe they were also preclinical, and it was a $2.7 billion deal with Kite. So it's something that it's always on our minds. And again, I think because we now have this platform, and can we find others in the space that have the CARs that we might consider combining together? Can we attract co-development funding for those things? I think it's a very important part of our business strategy moving forward.
Thanks, Michael. The next question is a general one, just around is there any updates on the rest of the portfolio?
Yeah, I didn't want to talk too much about that, obviously, in this presentation, because it is a bit standalone. But I think over the last few months, we have had good success with a number of our platforms. I think specifically as Ulpamest, where we signed on our Australian partner, Starter, for distribution of the drug in Australia and with an option for New Zealand. But also we had approval from the Ministry of Health for TEIDA covering the territory of Chile. So again, making good progress. And for our other other programs with Strides and Sanofi also making good progress on the reformulation efforts, which is great.
Thanks, Michael. I'll just pause for about 20 seconds and see if any further questions come through. Michael, there's no further questions, so I'll hand it back to you to give a closing comment.
Great. Thanks, Matt. Thanks again to NWR for organising the webinar. But again, a big thank you to everybody that has taken the time to dial in. We do appreciate it, and looking forward to giving updates on the platform as we progress for both the CD9 targeting therapy and for DKK1. Thank you.